Okay, welcome back for day 4. We have a kind of an interesting proteomics tutorial today. There are various kinds of tutorials we might try to go for, but today I'm going to try to introduce you to a couple of different repositories. Oh, that's drill. We're going to try to look at ways that you can acquire data sets of your own to work with from a couple different repositories. So I hope you'll find this interesting as we proceed. If you can't hear me from where you're sitting, feel free to move up to the middle chairs. And if not, just listen to the recording, because hopefully I'll sit near enough the microphone that it can pick me up. Okay, so we're going to be sampling repository data for proteins of interest. So I wanted to start with the fact that we have an awful lot of research in our department focusing on Parkinson's disease. We often focus on tuberculosis, but Parkinson's disease gets a lot of emphasis as well. So let us imagine that you, as a new graduate student, have joined a lab that does Parkinson's research. We would like to consider whether proteomics has value to add to the genetics work that we're doing. Now, maybe you're not ready to do a proteomics experiment yourself and want to see what's out there that you could use. So have other people produced proteomic data sets that we could use as a starting point for us to figure out what's reasonable to expect from our first proteomics experiment. So we're going to start at an indexing service called OmicsDI, written by a friend of mine named Yasset up at the European Bioinformatics Institute. All right, so OmicsDI is relatively easy to use. Lots and lots of bioinformatics tools out there have followed this paradigm that if somebody who's used to Google can also use your interface, you've written a decent interface. So OmicsDI is not going to search the whole web. It's going to search biomedical repositories instead, which gives it a much more targeted flavor. Now, along our directions, we're going to go to the search blank, enter Parkinson, and hit the search button. And what I want to ask is, does this searching for Parkinson also include related terms like Parkinson's or Parkinsonianism? So let's do a quick try of that. Parkinson. Did I do it with a capital P? I did it with a capital P. That's fine. Capital P. You can see it already has a whole bunch of terms related to this. And I'm just going to click search and see what happens. Okay, so I see that Parkinson, it did pick up Parkinson's with an apostrophe S here. Parkinson without the in. Parkinson apostrophe S. Maybe not Parkinsonism, but it's okay. We've got quite a few data sets here. You can see that this is not a repository that is specific to transcriptomics or genomics or proteomics. It lists all of them. So just coming over to the side here, we see that there are almost 800 data sets we can benefit from, which means that even without conducting your own experiment, you can benefit from the work that many, many other labs have already put into this disease. Many people don't think about all of the data assets that are available to them to begin with. Most of these labs will be very happy if you cite their work again, even if the conclusions you reach from their data may be different from the ones that they reached. Okay, so let us return to this. Today we've been studying proteomics, and so we're going to focus on just the proteomics data sets. To do a filter on that basis, we just click the proteomics link over at the left side. Now we've dropped quite a lot. We started with 400 different data sets. Now we're down to 77. This is already pretty remarkable. I would state that 5 years ago, the field of proteomics was very indecisive about whether or not people should be required to post the raw data underlying their experiments. There's pretty much a consensus at this point, at least among the journals, that people who are publishing proteomics experiments should be required to make their raw data available to others. And that's why resources like this are suddenly growing to a much greater extent. Okay, let us now see. We want to, one of the labs here that investigates Parkinson's disease frequently does so by fibroblasts. Now, when we think of the symptoms of Parkinson's disease in humans, we generally think of things like tremors. We think about how neurons respond to the disease, not necessarily what how fibroblasts respond. Of course, getting nerve tissue from a patient is much harder to accomplish than getting a bit of blood, and from or a skin biopsy. And from those, you can achieve cell lines that you can create more and more copies of, fibroblasts being a good example. So we want to investigate fibroblasts from humans, but we want our search to get a little closer to fibroblasts without just going to, say, fibroblasts. So let us now specify that we're not interested in mouse models, we're not interested in rat models, we're interested in Homo sapiens itself. So if I click this, I see that once again my search space has dropped to about half. I had 77 data sets to work with. Now, by specifying on top of proteomics that we want only human proteomes, we're now down to just 36 data sets. So have fibroblasts been a prominent model among the cell types used in these experiments? So you could ask, do we have a tissue down here at our left that reflects the kind of model we want to work with? We see that 10 people have worked with brains, 8 have worked with cell cultures, some have said tissue was not applicable, some have worked with cerebrospinal fluid, not available. Fibroblast, look at that. So there is a data set we can work with. So if you simply click on fibroblast, you should drop right back down to this particular set. Now, was this, given that somebody in our division has done fibroblasts, we might ask, who exactly posted this data set? I'm going to go ahead and click into it. Multifocal demyelinating motor neuropathy relating to P10. Okay, so this was submitted by Viet-Xuan Fan. We can take a quick look at the data set more closely, but we can also look down at the bottom there. The email address for Viet-Xuan is from isas.de. .de. Does anyone recognize what that extension means on an email address? Germany, exactly, right. So we can pretty much rule out that this person was working in South Africa. At least at the time they submitted it, they were probably in Germany. Great. Let us return to the set of questions here. So we emphasize that tandem mass spectrometry is a prominent technology for use in proteomics. Is it the only technology represented in these repositories? So we might start by asking that of this experiment, right? This is the one that was most directly relevant by showing up under fibroblasts. And it says it was collected on an instrument called an LTQ Orbitrap Elite. Okay, so we don't have an LTQ Orbitrap Elite in South Africa, to my knowledge. We have several instruments that are actually better than that, but the LTQ Orbitrap Elite is a somewhat older instrument than like the Fusion that we have over at CAF. The Fusion's a pretty recent instrument. But we might want to learn a little bit more about how this experiment actually operated, and for that we could go chasing into access data. For now, I will just specify that if somebody was using an Orbitrap, it's quite likely that this experiment was built around protein identification through LC-MS-MS. So if you see something like Orbitrap or another thermo-type instrument, it's very frequently the case that it was used for LC-MS-MS. All right, so let us back up one. We're going to undo this filter that we're only caring about fibroblasts for now. So I'm going to uncheck that under tissue. More generally, do we see that mass spectrometry is common or uncommon among the proteomics data sets that we have? I'm going to get the frequency breakdown by looking at technology type. You can see that a variety of terms get used here. We see mass spectrometry. It's not necessarily the case that just because a mass spectrometer was used, that these are protein identification data. So that's kind of a generic term. Someone may have been using MALDI-TOF, for example, or someone may have been using like MALDI-TOF profiling, or someone may have been using a triple quad if they were doing a quantification experiment. So mass spectrometry is a broad enough field that we can't just assume that people were building shotgun proteome inventories by doing so. However, we can see that 16 people specified that they are using shotgun proteomics. Shotgun proteomics is shorthand for what we talked about this morning. This is to say the sampling of tandem mass spectra that represent the fragments of individual peptides collected by whichever ones had intense signals in MS scans. We use the shorthand term shotgun proteomics for that. We also see people have used the term bottom-up proteomics. Now, you may remember that I spoke this morning about top-down proteomics being a type of proteomic experiment where we do not digest the protein before it enters the mass spec. So, in contrast to this top-down model, where you look at the protein as an intact molecule, people often speak of a proteomics where you identify peptides and from them infer proteins as bottom-up proteomics. So, bottom-up proteomics and shotguns and data-dependent acquisition proteomics, these are all synonyms for the same thing. Now, finally, there's an example here called Affinity Purification coupled with mass spectrometry proteomics. Now, I believe you've probably already had some exposure to Co-Immunoprecipitation. Co-IP? Does that term ring a bell? It does not. Okay, so maybe it hasn't been covered in this honor stuff yet, but it will come, especially for people who are studying immunology, I would expect. Okay, so the idea here is that we have an antibody to our favorite protein, right? So now we see YFP in these conceptual diagrams. YFP is your favorite protein. Let us say we're studying Parkinson's disease. We might be very interested in parkin, which is a gene that expresses a protein that is an E3 ligase. So in this case, YFP is this parkin E3 ligase. So let us say that I have an antibody that I can use to pull out parkin. That's great, right? But it may be that parkin only acts in concert with other proteins. And sometimes we find that proteins that interact together form this stable binding interface that if you're using very gentle techniques, if you yank out your favorite protein, another protein comes along for the ride. And you can use this to say, these two proteins are working in concert. Right? So yanking one protein, you get its binding partners and thus learn about protein-protein interactions. Protein-protein interactions are a very big deal. We'll be talking about those a lot more on Monday when we discuss biological pathways and networks. So you can see that 3 people have conducted studies here where they're looking at binding partners in association with proteomics of Parkinson's disease. Great. So that's not a whole lot of clicks, right? We didn't have to do a lot to discover the diversity of data sets that are out there that we can get for free. Grand. Did I have any other questions I wanted to ask? Oh, yes. What range of dates do these studies represent? Just the proteomics ones within Parkinson's disease. First off, how are we sorting? Relevance. I'm always a little suspicious of relevance. How does it judge relevance? In this case, I've given it one search term, Parkinson, so presumably anything that contains Parkinson is relevant and anything that doesn't contain Parkinson is irrelevant. That would be my guess. But I'm not really sure. So instead, I'm going to sort these by their publication dates. Now that doesn't necessarily mean the order in which they were run. It means the date in which the publications appear in the peer-reviewed literature, but that'll be close enough for this. So I've now sorted on publication date. What is the earliest study found in the OmicsDI index? There it is, 2006, November 30th. And then there's this really big jump all the way up to 2011. Now, I think I mentioned that it's worth paying attention to what the Human Proteomics Organization has to say about things. And one of the reasons why I like them so much is that HUPO was one of the very first big organizations to truly embrace the idea that if you do a big study, you need to make a big data set available. So they produced a brain proteome project way back in the middle of the first decade of the century, and all of those data are now available in PRIDE. But if you look at most of the published literature from published proteomics literature from 2006, it's impossible to get those raw files now, the raw data upon which those studies are based. From time to time, I send out an email to somebody who published a major study from the early 2000s, and those data are simply gone. Maybe they exist on a backup tape in a box somewhere in a closet of a laboratory that they've moved away from, right? The data might as well just not exist at this point. So proteomics has lost a large part of its history because people did not post these files to a large repository. So you might think, gosh, how irresponsible those folks are. You might just think, that was just a waste of taxpayer money, right? Well, I would point out that I myself moved away from Vanderbilt University in 2015. Are all of the resources that I had on my servers at Vanderbilt still available to me now? Big nope on that one. The machine that used to be my web server, that used to be our source code repository, is now running as a file server over in the proteomics lab. All the files that were on it from Vanderbilt have been erased. So things like the source code history from the early development of the ProteoWizard project may not be available now in any sort of accessible form. Scientific history gets lost all the time. You as a student can see that your data outlive your graduate training by seeing that they get posted to repositories when you publish. It's a big deal. Okay, so 2006, and are there really recent studies that have also been published? I'm going to zip all the way up to page 4. Oh, right, I could do descending, that's true. Good point. Okay, and I see that they run up to 10/24/2018. Well, we're now about 6 months beyond that, so maybe somebody hasn't published a really fresh proteomics data set for Parkinson here, but you know, the day is young. Okay, great. Are there other things I wanted to say? A fun one. Which of these data sets would you feel prepared to reanalyze based on the corresponding lecture? See, there's always this problem of translating a lecture into the skills you would need to reanalyze a data set. But I want to tell you that if you think a proteomics data set has valuable starting points for you in your graduate project, it is okay to talk to me or people in my group to help you get those data sets analyzed such that you can compare the gene symbols they have to the gene symbols you find of interest. If you think there's a data set out there that can support your research, it may take me a week to get the analysis thrown together for you, but it's something that we can do without a huge amount of work. It's a lot more difficult for you to get up to speed to be able to run all of the scripts and tools that we use on your own computers. You may not, for example, have a Linux box ready to go, but there are people around here who can help you get up to speed on these projects very quickly. Okay, with that, I'm going to move ahead to Panorama. Okay, so we were looking at OmicsDI, which is a really broad search engine designed for surfing a whole lot of different repositories. Panorama is a very different one. In the part of the lecture that I was not able to deliver this morning, we were going to discuss quantitation, specifically targeted proteomics experiments. A method that we frequently use for that purpose is called selected reaction monitoring, and one of the most popular tools for creating SRM experiments or for analyzing SRM experiments is called Skyline. So Skyline has become so popular that it has its own repository now called Panorama. You can kind of see how these terms go together. The Skyline of Seattle, the Panorama of Seattle. I lived in Seattle, so these terms, when they say Skyline, I of course am going to think of Mount Rainier off in the snow-capped Mount Rainier off in the distance. I used to be able to see it on a clear day, rare, in Seattle, but alas. Okay, long time back now. Haven't lived in the city for 19 years. All right, so we can see that there's a whole lot of stuff here. One of the really valuable things about Panorama is that they've developed a whole pile of training videos to help teach people how to design their own experiments and how to analyze their own experiments in this software. It's kind of rare that you can just go to YouTube and punch in a software name and get back a whole bunch of training videos, right? So that's a very valuable thing. To follow our directions here, I've laid out a kind of scenario where people would use this. Now, when you do a targeted proteomics experiment, you are typically narrowing your focus very tightly. In a discovery proteomics or shotgun proteomics or bottom-up experiment, you might be identifying 1,000 or 2,000 or for that matter 10,000 proteins from a sample if you're going really deep. But with targeted proteomics, you have to have a much more concentrated focus. Most typically, the rule of thumb that I use for this is that you can have 50 proteins that you think are really worth tracking down within this sample. So if you have 50 proteins for the sample, you're going to want to measure different peptides for that protein. So in a final assay, you would really want about, let's say, 3 peptides for each protein. So we started with 50 proteins, therefore we have 150 peptides total. Next, you're going to need a particular combination of precursor M over Z, the mass to charge value of the peptide, and you're also going to want a fragment ion M over Z value, a mass to charge value. So we have some peptide that we're going to blow up to produce a certain fragment. And each of these combinations of a peptide M over Z and fragment M over Z together will represent what we call a transition. So if you have 50 proteins, that becomes 150 peptides. And if you have 3 transitions, 3 fragments you're monitoring for each peptide, that's 450 transitions total. Each of these is going to be represented by a pattern of intensity as a function of time. Most of that chromatogram should be very uninteresting. You should just see the signal crawling along the baseline, and suddenly a bit of that peptide elutes from the column. And so you see the intensity rise very rapidly, and then it decays slowly back to the baseline. That's an ideal chromatogram. So the higher the signal you get from that sample, the more of that peptide you assess there to be. Now, because this peptide is showing up at a given time, all 3 of these transitions that we've predicted for it should rise and fall together. It's frequently the case that in a complex mixture, you will see other peptides that are the same combination of precursor mass to charge and fragment mass to charge. So instead of having this nice smooth baseline and a single rise and fall for this chromatogram, you'll frequently see that that chromatograms may individually rise and fall. But hopefully, if you've picked really well, all of the transition chromatograms are going to rise and fall together to add to your confidence that this is where your peptide of interest appears. Okay, so that's the general aspect of one of these targeted experiments, and a lot of that gets captured when you create a Skyline document. If you publish a study with Skyline, it's very kind of you to include the Skyline document in a repository like Panorama. That means that you can not only share exactly which transitions you looked for, including which peptides, which fragments you monitored for those peptides, but you can even incorporate within those files the chromatograms that were read out of your mass spec data. So you can share your experimental design very concretely, very exactly, and even the experimental results from it. So Panorama is quite good for that. Now, we are going to look at quality control. A lot of people have pinged proteomics repeatedly for being an irreproducible science. A lot of this came from the notion that if you run the same sample time after time after time on the same instrument on the same day, every time you run that experiment, every 90 minutes, you're going to get a different list of peptides back. Well, isn't that vexing, right? You got 1,000 different proteins in this sample. There are far more peptides that could be observed than you will observe in any one experiment. And that means that shotgun proteomics is a sampling technology. Of all the possible peptides, you're just going to see a relatively small fraction of them. One of the benefits of going with a targeted proteomics experiment is that we can greatly reduce the degree of variability that we see from one run of the experiment to another. Laboratories who've done really exhaustive studies where they all have the same sample and are running them on different models of instruments in different places have found that you can reduce the variability of targeted proteomics experiment to a coefficient of variance of, say, 12% or less. That's not terrible. That's still a good bit of variability among the results, of course. We'd love for that value to be 1%, but a 12% variability means that you can use these for finding relatively small differences. Remember, from difference testing, the lesser the variability of your technical variation, the better your ability to find small biological differences reliably. Okay, great. So we are going to go to a quality control example using data from the SPROCOP tutorial. People have created software just for doing quality control in the context of these experiments. Hopefully the link is still live. What's going on here? Okay, great. Now we have a series of questions that we wanted to ask about these. So while that loads, I'm going to take a look at this. All right, in this quality control example, they are using a study design to look for 7 peptides. Now, if you were looking for 7 peptides in a 7-peptide mixture, that would not be a great feat, right? If you were looking for 7 peptides in a biofluid, like serum or plasma, that would be a much more impressive kind of accomplishment. Because biofluids like serum and plasma have so many different peptides that your sample is swimming in that it becomes quite difficult to find a particular peptide unless you've got a really ace assay for doing so. Okay, so what kind of instrument did they use to produce this data set? Well, we can see that they've already given us quite a lot of specific meaning. We have individual peptides here that are spelled out, but I want to know a little bit more about the instruments that the instrument that they've used. I'm going to click on the sample files to see what I get. You see that these file names all contain the name a Q-exactive. A Q-exactive is a really popular model of mass spectrometer. There's a Q-exactive already in place at the Center for Proteomic and Genomic Research near Grooteskuur. There's another Q-exactive in the Jonathan Blackburn lab there. We have several of these around the country already. So I can tell you there's one over at what used to be called KRIF, over in Durban for that matter. So South Africa's got a fair number of these in operation. So in this case, they did not use an instrument that's kind of limited to doing quantitative style experiments. You could equally well use a Q-exactive for a shotgun experiment where you were just trying to say what proteins are here. But in this context, this instrument has been used specifically in a quantitative profile. Okay, I'm going to back up to that prior screen. Okay, over how long an interval were these data acquired? If they're doing a QC experiment, how many time points over what period was this experiment run? Does everyone see where to grab that? If you look at the citations on the graph here, it's pretty clear. So we see that the most recent of these was run in August of 2013. The very first one was run at the start of August 2013. So this is a pretty narrow fraction of time. We think of this as what's called a longitudinal analysis. Longitudinal. So this span of time is relatively short, but what we see is that the health of this instrument was profiled several times a day. We had, well, maybe not that frequently, but the 9th, the 12th, the 14th, and then the 15th, the 15th, the 15th, the 15th, the 16th, 17th, 18th, 19th, 20th, many times on the 21st, many times on the 22nd. So in looking at this time series, we'd probably want to realize very early on that it's visualizing it sample by sample or shot by shot, not proportionally by date. Otherwise, you'd see a whole cluster of these shots on the 21st and a whole cluster of these shots on the 15th. Okay, what can we interpret about it? Okay, so it's showing a Levy-Jennings control chart. So they represent a way to show the movement and regular measurements in the context of overall variation. So they are using a univariate analysis to assess how this instrument is doing. So one of the key concepts that shows up in connection with chromatograms is called retention time. You can see that the vertical axis here has been labeled minutes. So this minutes is telling us how long into the course of this experiment did the peptide ATEEQLK appear. Now, we can look into the files and find out exactly how long they are. Typical experiments are like an hour or 90 minutes or 2 hours, something like that. So the peptide ATEEQLK is coming off at about 15 minutes. So think about what we learned about chromatography this morning. Chromatography is not a mass-to-charge based technique. Chromatography, in the case of what almost everyone uses with mass spectrometry, is built around hydrophobicity. We have these beads that have long carbon chains on them. So peptides that stick really well have long retention times, and peptides that stick less well come off more quickly. So let's look at this peptide ATEEQLK. What is hydrophobic about that peptide? You remember the structures of amino acids? Oh, well, that's going to be what it is. What is a E? Does anyone remember what E stands for? No one's committing to this. Does somebody want to say it out loud? Well, I can't hear it over the drill anyway. It's glutamate or glutamic acid. Glutamic acid. So something to remember is that acidic and basic residues are not that hydrophobic. So the fact that we have two of those letters are already given over to acidic residues is probably a suggestion that this guy is a little less hydrophobic than he could be. In fact, there's only one amino acid here that's really strongly hydrophobic, and that is leucine. Leucine is a branched hydrophobic, so it's pretty sticky for those beads. But the other residues are not. The other thing I'd point out, it's a short peptide. Short peptides in general stick a little less well than do long peptides to these columns. Okay, so you can learn a fair bit about how this instrument is proceeding. I would note that the vertical scale on each of these graphs is not the same. Here they're showing a range of only 13 to 17 minutes for that first one. You can see on this axis here. On the next one, they're showing 25 to 50 minutes. So that has the effect of causing this second plot to look very compressed. The normal variation is might look just as big as it does on the top graph. But the reason why it's so compressed is that it's got this one point right here that looks like a really significant outlier. Right? So it's got one place where instead of showing up at about 33 minutes, that one time it appeared more than 50 minutes into the experiment. And you can see that they've layered what we would call metadata on top of that. You see this green X right here? They've labeled that instrumentation change. What if the operators of this instrument decided that it was looking a little weird, or that this column had aged a little bit, and they decided to change the column being used on the instrument? Well, maybe that first shot is going to be a little weird in its response. So we can often find that instruments perform differently just after a service. We want to believe that the instruments do not affect the variation in instrumentation does not affect the result that we get from them, because we're very dependent on instruments in biotechnology, right? But the reality is we should always be aware that instrument drift is something that is a reality we all deal with. Okay, it's good to track how different the instrument is being over time. All right, now I'm going to skip past this other stuff. I'm going to move past Panorama as well, because this last part is really cool, and I would really like to show you how to do this. We're not talking about protein structure in the bioinformatics section, and I'm really sorry about that, because it's a neat topic. But I would submit that on occasion you need to use structural information. We don't often think of protein structure as a systems biology, but in fact there's an awful lot to be learned from it. Have you had a lecture from Colin Kenyon? Colin Kenyon, okay. He's my next door neighbor. He's right down the hall here. He is an expert in protein structure stuff, and he has all kinds of interesting things to say about it. So if you ever have a question about why am I seeing an effect by a change in an amino acid that's positioned here, he's your guy. Definitely your guy. Okay. So let's take a much more mundane take for a moment on protein structure. Let us imagine that you must create a poster. You must create a poster for a conference. This is going to happen to you at some point. Creating posters is something that all sorts of people struggle with because they think, my goodness, what's visually appealing about what I'm doing? One of the things that you can always do is find some protein structure that relates to what you're doing. Protein structures are cool because they are 3D, and you can do all kinds of spiffy things with them. I wanted to show you just a couple of tricks to get there. So we are going to try to use a visualization tool with a structure retrieved from the RCSB, which is one of the sites of the Protein Data Bank, the PDB. So here we are at RCSB, this mirror of the Protein Data Bank. First off, I want to point out that they have some really cool articles about structure here. Every month, they have another molecule of the month, where they tell you a little story about how the structure of this critter allows it to do its thing. I think that's awesome. What a great idea. Okay, so we are going to come up to the beta-lactic globulin story. Right. I'm actually going to skip all the way down to the chymotrypsin. All right, so we're starting at the RCSB page. Once again, we are going to need an accession number. Accession numbers are the standard way that you get a particular data set out of the repository. In this case, the repository contains a structure for chymotrypsin. All right, so where do we enter that? Right here. We put a PDB ID in here. One AFQ is short enough that you can include it right there on your posters so everyone knows exactly what structure you're doing. All right, and now we have reached the crystal structure of bovine gamma chymotrypsin complexed with a synthetic inhibitor. So you might say, oh, that's already a cool rendering. I'll just grab that image. That is an amateur trick. We're going to learn how to customize that image to make it dance to our tune. So let us now go to the 3D view. We want to use JSMol to do this. Okay, I believe I have to click on this guy. Sorry. Oh, okay. I'm sorry. I need to do the structure view here, I believe. The interface changes very slightly from time to time, so I always have to do a quick fix. Okay, I am in a 3D view. Are other people able to get there? I clicked on the structure view to get here. All right. This first step I just think is fun. Grab onto something by holding down the mouse button and give it a yank. Woo! There you go. Right, so already we can do a 3D manipulation of this structure by rotating it around. You can see we can get little bits of information. See, there's this weird bit going on right here. See that little dude hanging out there? What is that? I see that this is SO4. So this is sulfate hanging out nearby. All right, different atoms, that's great. You can see that there are some structural elements that we can see here. Let's return to my direction. I get really excited when I do this stuff, so, all right. We are going to use JSMol to do this visualization. So, JSMol, that hammer, I swear, it's driving me crazy. Select a different viewer. We are going to switch over to the JavaScript molecular viewer, JSMol. It's going to give us a little more fine-grained control over what's happening here. It's going to think about it a little bit, though. Things are happening. Okay, there it is. All right, already our view has changed, hasn't it? So the default of how to visualize these things is a little different for this viewer than for the other. I understand you've got a schedule light. I got that. Okay. Oh, look at that. Something happened. I've now got what we call a cartoon view. What are some of the most common elements of secondary structure for proteins? Give me an A, give me a B. Okay, alpha helices and beta sheets, right? So already we can see that this visualization... Just get the door shut, get the door shut. Okay, see, have a good weekend. All right, so in this case, we have a visualization that is immediately making clear to us which parts of the structure are alpha helices and which are beta sheets. And yes, this one is just as tuggable as the other, so you can pull those around to get a better view of a particular area. Okay, so we are going to find a sequence we're going to take a look at the PDB file that underlies this. You know, all of these pretty visualizations, they're built off of some data type, right? So down here at the bottom, what was I looking for? Sorry. Near the bottom of the header, you should see the sequence of amino acids included. So, oh, I'm sorry, it's at the upper right. I lost track of where to find it. Okay, sorry. Under Display Files, you can click out that menu and get the PDB file. I'm going to look at just the header. I think that should be enough. We'll see. Okay. So this is the file format underlying all these PDB entries. So these visualizations are just a way to show what's already encapsulated here. Now I had some questions on this. Right near the bottom of the header, you should see something that says Seq Res. Okay, so I'm going to do a quick search for SEQRES. There we go. So you can see it uses kind of an old school way to represent the sequences of the polypeptides here. Cis, gly, val, pro, alla, illy, glyn, that sort of stuff. Right, so that information is all captured right there. Now, this might be where I get in trouble for using only the header. I wanted to see a line called site, S-I-T-E. Ah, good, it is still here. Excellent. So site has some interesting values for us that tell us about things like a catalytic site. Do people remember tales of the catalytic triad for serine proteases? Everyone had some biochemistry classes back in the day. Serine proteases. Catalytic triad? Good. Good. It was one of my favorite stories from biochemistry class way back in the day. So I want you to note that the people who made this structure have annotated parts of it to make it easy to find. And so here we see that the catalytic site has been marked as representing histidine in the B chain at position 57, aspartate in the B chain at position 102, and serine at in the C chain at position 195. Grand. That's going to be helpful to us in just a minute. Now, do I want to grab that? No, I don't. I want to just I want to step back now to look at the structure. We've looked at the data upon which it's based. Now we're going to come back to this structure, and we see that it has re-rendered itself beautifully for us. Thank you. Okay. We are going to script. We are going to do a little programming on how to visualize this structure. So we need to make this structure a little easier to work with. We're going to use something called a trace view under Style. All right, everyone see where Style is here? Oh, it's down, it's down. So this is the cartoon structure. We're going to alter this to be trace. Shoop. All right, see how cool that was? Immediately our beautiful little curly bits and anti-parallel sheets and so on, they vanished because they're just colored yellow now. And colored, the coloration of the alpha and beta helix structures are now in place. All right, we are going to expand the scripting options section. Hopefully they haven't changed our user interface entirely. Oh, sorry, I changed that to ribbon. I didn't mean to. Gotta come back here. Trace. Good. Okay, scripting options. There it is. All right, is that okay? I've clicked on that little drop down and now it's open like this. Good, good, good. Everyone able to get that far? Brilliant. You guys are faster than I am. That's good news. All right. All right. And the goal of every teacher is to see that his students are better than he is. Sheesh, I made a lot of mistakes along the course of my career, trust me. Imagine what you guys can do with good training. Huh? That's my hope anyway. I want to be a good teacher. All right. So the JS-Small language is reasonably intuitive. What was the last time you thought a programming language that was reasonably intuitive? I liked Logo back when I was in elementary school. They would teach us how to, do pen down, move forward, turn right 10, forward. That stuff was great. Love that. That was intuitive. This is not. All right. So we could change our background by saying background blue. Obediently, the software will say, aha, I shall turn your background blue. So if your poster color is blue, you can use that line. All right. But we want it back to white again. Background white. Did someone go red? Did it look okay? That's awesome. Okay. Many operations can be applied to a subset of the atoms on screen. We are only interested in this case in the catalytic site, right? Remember that was specified back in the file there? All right. So we need to specify that exactly these amino acids are selected. All right. I am now going to enter select 57, 102, 195. Those are numbers I took right out of the PDB file, and click submit. Okay, hopefully nothing happened. But something did happen, it just doesn't appear yet. So now we come back to this, and now we want to specify how those should be shown. We're going to change how just these letters are shown. Just these structures are. I enter this, poke that in there, and say submit. Now, when you pop back up here, you should notice something a little odd has gone on. So, you see that there's a little ball and stick arrangement here to show the side chains of these amino acids. None of the other amino acids have side chains. But because we selected those three and specified this type of visualization, just those changed. Okay. I'm actually going to try to rotate this a little bit so that's clearer. Shoop. Oh, look at that. They're all in close proximity. Something magical is happening. All right, remember this is the catalytic site. So these three amino acid side chains are all in close proximity, and that proximity lets them take on some actions that they wouldn't by themselves. Okay, great. Now, we next want to increase the contrast so that these stand out from the background. So this time we're going to say color red. All right, so we come up here. Color red. Boom. And kerpow, just like that. We've now drawn attention to just this part of the protein structure. Imagine how much better that's going to be when you make a poster, right? Now, just the part of the protein that you are proposing has a sequence variant that's significant will light up. That's pretty special. Great. Okay. Which amino acid is the histidine? Everyone know? What were the three amino acids we highlighted? Okay, serine, arginine, and histidine. So we could be really lazy and just move the mouse until it was over one of these, and it should just tell us what it was. See that? So histidine 57 is right there. But you might have remembered that histidine is the only one of those that has a loop. It has a cycle in its side chain. So here we can see that it's lit up for us quite nicely. Okay. So now you must show your own artistic sense because you're going to position this subject so you can take a snapshot of it. I think something like, oh, this one's tough. Maybe about like that will be good. Now we have the option of exporting this image to a file that we can easily use. You might think download files would be it. It's not. It's someplace else here. This is by file export. Oh, I remember how I did this one. Okay, sometimes you just need to be a crafty and old person to find the shortcuts here. I'm going to right-click the image. Oh, no, there it is. Okay, I thought I was defeated. I wasn't. Okay, when you right-click, you should get this little image of a menu, and I can export a PNG right here. So if I export a PNG, I'm going to call it JSmall ping, and the next thing I know, I should have a downloaded file. Ah, there it is. Now I can go into my software and paste that in and have my very own image of this structure for my poster. Okay, so I know that's not much of an introduction to protein structures, but it's some of the cool stuff, and I hope that you find it useful. Some of the structures like this can be easily found through Interpro. So if you have a sequence, but you're not sure what structure belongs to it, you might try putting it into Interpro, which will get you a gene 3D hit to a structure you can use. It's pretty powerful. Okay, so that was a very rough guide to what the repositories have for us and how to get splashy images for your poster. And with that, we are done for the day. I hope that you have a brilliant weekend and that no jackhammer is going near your window. All right, thanks guys.